Okay, so now I'm recording and I want to get into, where are we? So hopefully Zoom people are just seeing one big screen of the light reaction.
Yes, we are.
Thank you. So here is the familiar game of how the plants are doing the light reactions of photosynthesis. So we have worked through it from start to completion, looking at how water is the ultimate electron donor, NADP plus is the ultimate electron acceptor, and we have many players in the game that are handing off the electron from donor to acceptor on down the line. Remember, there's some added complexity when we get into the Q cycle that's being played out in cytochrome B6F. And so that is something to work through. What was the, so the point I was trying to get to there is that, remember, there are steps in this process where, I put down in the PowerPoint slide, repeat. And so we have to think about, that plastoquinone in particular, that 2-electron gate, that plastoquinone just isn't happy with 1 electron, it has to have 2. And so we do have to think about, okay, you have to repeat back to photosystem 2 getting hit with light energy in order to get another electron to satisfy the second plastoquinone to get 2 electrons, and then it can grab 2 protons. So there are certain sections like that you have to think about this repeating idea in order to satisfy, especially the plastoquinone. Jay-Z, you had a question?
So I know this won't be till exam 6, but on the exam, is it going to be okay if we just state, like, then repeat from photosystem 2, light hitting,
Yeah.
blah, blah?
Yeah, so like don't go through it all over again in written form. You can just say, and then this repeats back to photosystem 2 getting hit with light, done. And then you can move on, okay? So, and then the other thing that reminds me, I wanted, and I'll remind you again, because I know you're very focused on exam, all of you are focused on exam 2 appropriately so, but when we get closer to exam 3, if you want me to read out what you've typed up about the light reactions, I'd be happy to do that. I've had students do that in the past, and that really helps them make sure that they got it down, because I'm still leaning on doing an essay exam, an essay question on the light reactions to show. And if you've talked to anyone who has had my exam on the light reactions, it's a huge essay question. So I'm just warning you right now to get you serious about studying the light reactions.
You're going to do it as like one big question and not break it up into several smaller sections of questions?
Typically, I do one big question. I'll set the stage for you. I'll tell you exactly where to start and then what not to forget. And so I'll lay it out for you. But I encourage students to try, writing that out several times and then let me see a version of it before you actually do it for real. And that also will kind of force you to get comfortable with it before you actually take the exam. And so I'm really trying to do what I can on my end to make sure that you're comfortable with it. So when you actually do it for real, it's like, oh yeah, I know this. So that's coming up, not for this exam on Friday. That, by the way, the exam on Friday will start at noon like the last time. And I am going to, rather than just monitoring my email, I will have my Zoom up and running through the virtual office hours of this course. So if you need to Zoom in to ask me a question, that's probably quicker. And so I will be available during the exam to Zoom in. And so that's, please ask me any questions during the exam for clarification. All right, so we have been talking about the light reactions, and we looked at what's being shown here is linear photophosphorylation, which just means the electron path is linear, starting at water, ending up at NADP+, and during that whole process, you get a proton gradient getting established. And so the photophosphorylation just means that you have ADP getting phosphorylated to make ATP, photo through the light energy process, the light reaction. We also talked about cyclic photophosphorylation, which is this one. And again, the cyclic is just referring to the electrons. Certain portions of the thylakoid membrane the plant will decide, so to speak, I need more proton pumping going on. I'm getting short of ATP. I need to boost my ATP levels. So I'm gonna devote certain sections of my thylakoid membrane into doing the cyclic photophosphorylation. And that is basically, when you look at how they're running that, it's a circular flow of electrons. And when you look at the payoff is you get proton pumping. And so that allows for more ATP production. And so certain plants that have a higher ATP need, well, we're going to talk pretty quickly here about carbon fixation. And there are certain plants that do what we call C4, they're C4 plants. And they have to have a little bit more ATP because of what they've done to adapt to their environment. And so very often during the day, they'll do this. They'll go, oh boy, I need a little bit more ATP. And so it's all happening right in the chloroplast, so carbon fixation is happening. So it makes sense that they're going to kick into cyclic photophosphorylation, at least for a little while they can boost their ATP. Okay, so we talked about that. And then we talked about the idea that with the lab exercise that you did, DCMU, I think this is where we left off. The idea that when you were playing around with DCMU in the lab, carefully, because it is a herbicide, it really impacts the plastoquinone electron flow right there, from the first plastoquinone to the second plastoquinone. It's nailing it right there. And they know that that's what it messes up and stops the electron flow, and can mess up at certain doses. Dose makes the poison. If you dilute it out enough, it's minimal. So there are issues, both what we can do to plants to mess up the light reactions, but then if you just think about the plant's ordinary life, if they're hanging out in their habitat, and you may have heard the expression my mom used to say, too much of anything can be bad for you, and she is right, your mom's right, listen to your mom. So excess light energy certainly can damage plants, just like we get not just like, but sunlight damages us too. We get sunburns and possibly skin cancer. Plants don't get skin cancer, but they can still have issues with free radical formation, and that can damage. We get the same thing happening in our cells. It can damage the lipids and the DNA and the proteins. And so sometimes you'll see the situation where the plant is suffering. And so we call it photooxidative damage, because of course we have to throw these big words out at us and make us memorize it. So photooxidative damage is the fancy word to talk about it, but it's just too much light energy coming in too much at once. So, normally if the plant is getting hit with light energy, it excites the electron of the chlorophyll pigment in the reaction center. That's great. That's called photochemistry, and that's great for a plant. All right, that's what we've been talking about. That's a good thing. However, if it's too much light energy coming in where that reaction center can't keep up with the amount of quantum energy coming in, the photon, then it can start to cause chaos and problems. And so that energy will start affecting other molecules besides the chlorophyll pigment, and that can be very bad. And so toxic chemicals is kind of the umbrella statement we give to the certain chemicals that can get produced under these conditions. And an example would be singlet oxygen. So oxygen gets into an excited state, and I'm not getting into the physics and chemistry of it, but that can then start to, this whole idea of being free radicals, where they start to react with nearby molecules and compounds and can start creating this like domino effect of reactions going on that lead to lots of damage of, like I said, it can damage lipids, proteins, DNA. And so this can be a kind of a compounding domino-like effect. And of course, when you look at the chloroplast, right, you have the thylakoid membrane. If that starts to get degraded, and then of course all the proteins and pigments involved in the photosystems. There's a lot of issues that could happen that could go very wrong. So when they studied this and they started, playing around with it in the lab and, what happens if we hit the plant with too much light? What do they do? One of the things that they noticed that plants can do is use some of their other pigments, like the carotenoids. So yes, the carotenoids are accessory pigments that are helping to capture different wavelengths of light energy and getting that sent to the reaction center. So carotenoids are great for that, but they have more than that role to play. They can also do this. They can, what we say is they offer photoprotection. And some scientists say they're like a safety valve for the plant to absorb this excess light energy and keep it under control. So they actually use the word quench. The carotenoids can quench the excited state of chlorophyll. So what happens, and I'm going to give you the big picture idea here, is that the carotenoids don't get too excited when they absorb some of this excess light energy. They don't get overreactive. They can absorb it and they can run through a little reaction. And as they're doing that, it dissipates that extra energy, dissipates as heat, which is much safer than a free radical. I'd opt for the heat too. So and so they're kind of this nice, stable, helper, if you want to think of it that way, that takes that excess light energy and pretty safely converts that excess light energy to heat energy. And that's a better situation than a free radical formation. So let's look at a kind of a flow chart. When they did these experiments in the lab, they could see that, off to the right is what you want to happen. You want the light energy to be used for photosynthesis. So that's, what you want. But this flow chart down now is what can happen if too much light energy comes in. So excess photons, you can see this heat idea, that you get release of heat, and that's a safe way to deal with it. Here in this green box, you're seeing different kinds of free radicals. All right, so these are just some examples. And you're seeing a second line of defense. You can have carotenoids, again, playing a role. And then we're going to talk about some enzymes that can play a role. And we're going to talk about D1 a little bit, because they've done a lot of work examining D1, which is a protein that's part of photosystem 2. So let's take a look at some of these ideas that are on this flow chart. So carotenoids, what's going on? So xanthophylls are carotenoids. All right, so they've done some work in the lab where they see this reaction take place, where these are all carotenoid pigments. All right, so you have violoxanthin reacting to form an anthrozanthin, what they call an anthrozanthin type of carotenoid, which then reacts and forms zeaxanthin. So basically, it's just in this little reaction pathway, you get the dissipation of heat energy. So that's the payoff. It's like the carotenoids go through this little reaction, and as they're doing that, it's a safe way to just kind of release this excess energy as heat. I'll show you a visual of this if you're more of a chemistry brain person and like to see the chemistry of it. But again, the big picture is this reaction allows for heat dissipation. So it's a safe way to deal with the excess energy. So here you have the violoxanthin, and if you follow down, you can see some minor shifting going on here. And so they can actually measure this. If they start out the plant with low light, they'll see that there's a heavier amount of violoxanthin in the tissues, leaf tissues. And then as they start to ramp up light intensity and really bombard the plant with too much light, you'll see, you can measure this reaction going on and the uptick of zeaxanthin. And so they figured out that reaction pathway pretty easily in the lab setting. So that's one idea.
Quick question.
Yes, go ahead.
Were those carotenoids, are they donating some electron to that photosystem 2 P600?
Yeah.
80?
Or so these carotenoids are part of the accessory pigments. They can be found right with the reaction centers. and part of their job will be to absorb light energy and get it to the reaction center. But if too much light energy can come in, then you can see these pigments, the violozanthin, will start going through this reaction pathway. So they're right there in the thylakoid and the accessory antenna pigment systems. Yes. So they've looked specifically at the different photosystems, and they've done more work with photosystem 2, I think primarily because it's where the oxygen evolving complex is, and we're so fascinated by that and trying to still figure that one out. So they've done a little bit of work with this, well, more so I think than with photosystem 1. And they can experimentally show that if you hit, the chloroplast in photosystem 2 with too much light, the photosystem 2 will eventually break down into a non-functioning photosystem. And when they do these experiments in the lab, they can do it very carefully, and they can show, yeah, there is the ability of the plant to repair photosystem 2 with minimal damage, but then there's like a tipping point. And one of the first parts of the photosystem that it's almost like the weakest link idea, is the D1 protein, where the D1 protein, which is being shown here in the kind of the golden yellow, not the orange, but you can see there's some yellow on either side of this picture. That's the D1 protein. And so it seems like the D1 protein kind of gets sacrificed, and that's the one that part of the photosystem that gets damaged. And they can see that the plant can actually remove that and make a new one and slip in the new D1. And so there is this ability of the plant to kind of, do some surgery, so to speak, on the photosystem, which is quite remarkable. And so there are, and they're still figuring this all out. There's a lot of unknown about this, but at least we're seeing under, controlled lab conditions that the plant has this remarkable ability to repair on the fly, as it's getting, trying to continue to do photosynthesis. It can remove the D1 if it gets damaged and replace it with a newly synthesized intact one. But like I said, eventually there's a tipping point where if you keep experimentally hitting it with more and more light energy, it will completely break down. So that's some of the work they're doing with photosystem 2. Photosystem 1, they've done some work with that, and they can see damage to photosystem 1 as well. So remember, ferredoxin is associated with photosystem 1. It's waiting for an electron it gets an electron from photosystem 1. And so its job, normally, ferredoxin should give its electron to FNR, to the FNR protein. But sometimes that doesn't happen under too much light energy. Sometimes it'll accidentally, so to speak, give its electron to oxygen, and then you get a superoxide free radical. And so that's all happening right there by the thylakoid and by photosystem 1. So now that free radical could damage photosystem 1, it could damage the thylakoid. And so there are plant enzymes that the plant has in order to act on these free radicals. So superoxide dismutase, ascorbate peroxidase are two such enzymes that can attack these free radical oxygen species. And plants aren't the only ones. There are other organisms that have these types of enzymes as well. So.
Sorry, but a couple slides back, there was an acronym that was ETC. What does that stand for?
Oh, ETC, whenever I put that, especially in capital letters, it's electron transport chain.
Oh, okay.
Yeah, not et cetera, et cetera. So ETC is electron transport chain. So, okay, so they're looking at both photosystems and trying to see what can the plant do in order to try to protect itself or repair itself. But then again, too much of anything can be bad for you. If you keep pushing it in the lab situation, you can cause total breakdown of the both photosystems. So, okay, so plants do have these abilities to deal with too much light energy. And then the other big thing that they've been looking at is, okay, we know plants have two photosystems, Other photosynthetic organisms only have one, but plants are special, they have two. So we know that both of these photosystems need to get hit with light energy, and they need to get hit with the same, amount of energy, if you want to think of it that way, so that you can keep the electron transport chain process flowing constantly and continually from, getting electrons all the way from water to NADP plus. And so those two photosystems, I like to think of it like, a band playing a concert for you. It has to be done in concert, or a marching band. I used to watch my son play in the marching band, and, you have to be in step. You have to be playing together for it to be beautiful and make sense. And so they've discovered this really interesting thing that plants can do if things get out of step, so to speak. And so you want the same energy input at both photosystems so that the electron flow is continual and smooth and it's efficient. Everything's clicking along just right. Well, that's not as easy as it sounds. If you think about it, especially a plant, where its photosystems are very commonly are getting exposed to different light intensity. If you think about shading action, a cloudy day, the upper canopy shading the lower canopy leaves, you got all kinds of variables that can really mess with that. And so they've had to adapt, they've had to evolve mechanisms to try to deal with this. And one thing that they've done is that plants can actually shift the energy over to the photosystem that's lagging behind, that's not getting enough light energy. It can actually kind of say, here you go, here's some extra energy. That should allow us to go back into step. And so what they've done experimentally is they've noticed that there is an antenna complex. The LHC2 is light harvesting complex 2. That's way back in a previous slide. We talked about those. That's just that's a shorthand way to talk about the antenna pigments that surround photosystem 2. So light harvesting complex 2 is photosystem 2's antenna pigments. What they've noticed experimentally is if those proteins that associate with that antenna pigment complex, if the protein is phosphorylated, so that little squiggle P means it's phosphorylated. So, and if you've taken other classes, this may sound vaguely familiar. It's very, it's typical across all types of living organisms that phosphorylation is a way to signal, to communicate that something needs to happen. And so this happens a lot, even down into the microbial world, we see microbes doing this as well. This phosphorylation is a way to communicate something's got to happen here. If the LHC2 protein is phosphorylated, that tells the antenna pigment, I need to deliver this energy I'm absorbing to photosystem 1, even though normally it's associated with photosystem 2. Okay, that's what's so weird. It can physically move, these antenna pigments can move to photosystem 1 through the thylakoid and give that energy to the photosystem 1 reaction center. So it's this way of compensating where if photosystem 1 isn't getting enough light energy, this phosphorylated form of the antenna pigment can move and associate with photosystem 1. If that antenna pigment complex is not phosphorylated, that's also telling the plant, oh, okay, I'm just going to keep the LHC2s around photosystem 2 and just keep doing that. Everything's good. Now, there has to be a way for the plant to say, it seems like photosystem 1 isn't getting enough light. There has to be some process in place, and they're still working this out. But one thing they have discovered is that it does partly depend on the amount of plastohydroquinone and the concentration of that.
What does getting energy from photosystem 1 to 2? Depends on that.
Where LHC2 will deliver energy.
Okay.
is dependent on PQH2 levels. So now the plant needs to assess how much PQH2 do I have in the thylakoid. Remember, the plastohydroquinone is part of it's a player in the game. And remember, the plastohydroquinone, it's that 2-electron gate. There's the first plastoquinone, which gives the electron to the second plastoquinone. Then all of that has to repeat. Eventually, that second plastoquinone gets 2 electrons, grabs 2 protons from the stroma, and releases. And then PQH2 has to go to cytochrome B6F. So that's the portion of the light reactions we're thinking about. So let's think about that for a second. If PQH2, if the levels start to get high, if PQH2 starts to build up, what that's saying is somewhere down the line, there's something wrong. PQH2 can't do its job of donating off an electron. And so PQH2 is like building up, and he's probably yelling down, hey, Bob, pick up the slack, let's go here. And so photosystem 1 is slacking in terms of not enough light energy hitting photosystem 1 to keep this going. So it's like it got backed up, and it's the fault of photosystem 1 not getting enough light energy. So PQH2 starts to build up, going, I can't do my part until you guys kick it into gear down the line here. All right. And so if we go, let's just look at the top picture here. So plastohydroquinone right there, that if that starts to build up, what that's saying is everything downline from it, so to speak, downstream of it, is not doing its job. All right. So what that means ultimately is photosystem 1 is not getting hit with enough light energy, and it's causing, all of this part of the light reaction to slow way down, which causes PQH2 levels to just, build up. Photosystem 2 is still doing its job, so it's making a lot of PQH2, but it can't do its job of delivering off an electron because photosystem 1 is slacking or not getting hit with enough light energy. Does that make sense? Okay. Yes, go ahead.
How does photosystem 1 and photosystem 2 being arranged differently in like the grana versus the lamellae, I want to say, affect the light harvesting complexes moving from one to another?
Yeah, and it's hard to wrap my head around, but they can demonstrate that is happening. I don't remember the exact procedure they used, and you're right, because photosystem 2 is found more in the grana area, the stacked region. Photosystem 1 is more in the stromal lamellae region, and yet you can still see these antenna pigments shifting laterally. So it works out well enough where they can see this shift going on, and at least so far, they know one thing the plant is paying attention to is the PQH2 levels. That's kind of telling the plant, oh, things aren't, if you get really high PQH2, then the plant has to make sure that photosystem 1 is ramped up a little bit, and let's get you more energy so that we can get back in step and do our electron flow more efficiently.
So it would make sense for this to happen more towards the edges of the grana.
Yes, because they're physically closer to each other. You're correct.
Well, that, and also there's going to be more PQH2 by the photosystem 2 stacked in the grana region, just because there's more photosystem 2 there.
Yes, you're correct. And so it's they're still working on this, but this is one of the first findings that they could verify experimentally, that high PQH2 really is a signal to the plant. And it gets messy, like you say, JZ, because of the thylakoid. The distribution of all the transmembrane complexes is not even, and it, the way we talk about it, it doesn't look like this in the thylakoid membrane. It's just for ease of talking, we talk about it as if everything's right next to each other, but they're not in reality. So that kind of makes things more complicated for sure to think about and experimentally measure. I like this. I just put this in here because some of you might be actually more familiar with aerobic respiration. of what's happening in our mitochondria if you've taken animal or human phys. And I just love the beauty of how similar this is. And so often when I interact with juniors and seniors who've had enough of this foundational knowledge and they start to piece together, oh my goodness, and, look at how similar some of these pathways can be amongst different organisms. And it's really quite beautiful when you look at the similarity here. And so if that also helps you to think about the light reactions, if you can use what you've learned in previous classes to lay the foundation, so to speak, to help you process this new information, well, that's a good strategy too, as you're learning the light reactions. Okay. So a remarkable process, splitting electrons, splitting water to get electrons is just amazing. And, the ability to do that, you need quite a bit of light energy. And then ultimately you end up with NADPH and ATP. So now we have to talk about, okay, so what? Now what? So now we have to get into the idea of once the NADPH and ATP are made in the stroma, now we could talk about part 2 of photosynthesis. All right, so that's the next slide set. I believe they're available in EDUCAT. So let's look. And
Weird question real quick.
yes,
There's like a lot. There's like NAD plus and NADH and NADPH.
yes,
NADP plus and like.
yep. Depends on the organism you're talking about, but NADH and NADPH, the P is a phosphate added on to that to the NAD,
Yeah.
and it just depends on the organism which they're going to use. Higher plants like to use they'll make NADPH, but if you take a microbe class with me, you'll find some microbes prefer to make NADH.
Okay, but they do like the same job.
Yes.
Okay.
Yes, they're both. NADH and NADPH are both very happy to give off electrons to someone else now. So that's what we got to focus on. We got to look at what good does that NADPH do for the plant. They're so busy making it during the light reactions, but so what? What are they using it for? Are the Zoom people still seeing just one big slide?
Yes.
Okay, thank you. So here is a big picture overview of the whole everything of photosynthesis. And we're going to now zoom into the side on the right here where it says the Kelvin cycle. Depending on who you talk to or what you read, people may call it carbon fixation. I tend to say that a lot. I'll also talk about the Kelvin cycle or the C3 cycle. So what I tend to say is on the top. I can't remember if your current book is using the one on the bottom, photosynthetic carbon reduction cycle, but that abbreviates that down to PCR. And when I say PCR, what do you think about?
Polymerase chain reaction.
Yeah, polymerase chain reaction. So it's like, okay, that's confusing. So I don't use this bottom jargon. I don't do that. So you're going to hear me say the Kelvin cycle, carbon fixation, C3 cycle, all right, those terms. So Kelvin it's named after Dr. Melvin Kelvin. That's his real name. I imagine he might have got beat up on the playground when he was younger, but his claim to fame is he went to Michigan Tech University. That's where he got his bachelor's degree, and he won the Nobel Prize years later, for his work on carbon fixation. So that's why it used to be just called the Kelvin cycle. Now it's called the Kelvin-Benson. It's CBB. I can't remember the other B. 3 different people, technically, I can't remember. I'm so used to just saying Kelvin because it's easy to say it that way, but there's actually other scientists that were involved, and I'm blanking on the last one, Benson, and can't remember the last B. But anyways, overview. So what we just talked about is being shown on the left here, photosystem 2 and 1, the chlorophyll pigment absorbing light energy, and you get NADPH and ATP. And this is a nice visual because it's showing how once that happens in the stroma, you start kicking in then to using the NADPH and the ATP to push the Kelvin cycle. Finally, you see the CO2 coming in and getting reacted to form carbohydrates. So remember, I don't like you using C6H12O6, abbreviate it down to CH2O within parentheses and a subscript N. is the better way to go. All right, so the Calvin cycle, C3 cycle. They named it C3 because that's the first stable molecule, but it's not the first molecule. It's the first stable molecule. So this confused them early on, years and years ago, when the technology wasn't there to really get at this immediately. It left them really struggling with, okay, we know CO2 is coming in. They figured that out. CO2 is coming in and getting used to make carbohydrate, but what does it react with? And so they know the first stable molecule is 3 carbons. Carbon dioxide has 1 carbon, so they said, oh, let's look for a 2 carbon. It's a 2 carbon molecule that CO2 is reacting with. And so they searched and searched and searched. Came up empty, and then Dr. Melvin Kelvin said, well, there's more than one way to get to 3. What if you go up to 6 and then come down to 3? You know, 6 divided by 2 is 3. Of course, he didn't just do that. He did a lot of work. But basically, he helped them look a little differently at this idea. And they found that, yes, it is a 5 carbon molecule. It's a 5 carbon sugar that CO2 reacts with. And there's this very unstable intermediate that's 6 carbons. But then that 6 carbon molecule gets cut in half, so to speak, to 2 identical 3 carbons. So it was tricky, especially back then. The technology was so limited. So the name of this sugar is ribulose 1,5-bisphosphate, which you can just abbreviate down to RUBP. All right, so this is a sugar. Ribulose 1,5 means there's 2 phosphates at the 1 end and the 5 end of the carbon molecule. Bisphosphate, 2 phosphates. Okay, ribulose 1,5-bisphosphate. So the CO2 will react with that sugar. And so once they figured that out, that was one of the big stumbling blocks. And then they were able to start tracking from there. They already knew that, you know, the first stable molecule was a 3 carbon. So they just had to figure out what did it react with. Yes.
When I was younger, I was taught that like sugars are molecules with carbon, hydrogen, oxygen, and like a 2:1:2 ratio. But that's obviously an oversimplification. But like, do you have a definition for like what makes a sugar a sugar? Or?
Well, the CH2O designation, I guess, is how I always think about. That's like the basic building block.
Yeah,
so
that's not like 1:2:1, but then that doesn't work once they polymerize. So as soon as you have like sucrose, then that's not the ratio anymore.
Right.
I don't know if that's even a valid question. But.
Yeah, I don't know. I mean, I guess I'd have to look to see how the biochemist would define it. Once you get beyond, like you said, once you get beyond the basic building block. It's a good question. This one I'd like you to get familiar with, please. This is like a big picture idea, and I will talk about it on the exam as kind of like, this is the pie chart. So if I reference, think back to the pie chart of carbon fixation, this is what I'm talking about. And so they break it down into 3 different sections. So the first one we'll talk about is carboxylation, and then there's a reduction phase, and then there's a regeneration phase. And so this is the pie chart, big picture idea of the carbon fixation, the C3 cycle, the Calvin cycle. And then I put in another visual very similar to this. So you see here in part of the reduction, you see this offshoot. So the Calvin cycle is the big green swooping lines. It's a cycle, but there's an offshoot that we need to consider too, and a very important offshoot, because that's where you finally get sugars being made, all right? So this offshoot we'll certainly talk about, and then in this next image, they just kind of beefed that offshoot up and gave a little bit more detail. So it's the same image, it's just a little bit more detail in this offshoot part that is showing a little bit more detail about sucrose, what's going on with that, what happens when the plant wants to make starch. So we'll get into all this, all right? That's part of the process. So let's start with phase 1. Carboxylation. So that just means you're adding a CO2 to a molecule. In this case, it's going to be RUBP. So let's write that up. So if I were to ask you on the exam, tell me what the first step of the Calvin cycle is, this is it. It's CO2 reacting with RUBP. You get a very temporary 6-carbon molecule. I don't even really want you to worry about it. The big picture idea is that you end up with 2 molecules of what we call PGA. And PGA stands for phosphoglyceric acid, and it's phosphorylated on the 3rd carbon, so that's why it's 3 PGA. So, but you get 2 identical molecules of PGA. Another way to say phosphoglyceric acid is phosphoglycerate, if you want just a one word, phosphoglycerate, but it abbreviates down to PGA, and you can just use abbreviations. You don't have to write it all out. The enzyme that catalyzes this is a very, very special enzyme called Rubisco. Tons of research going into this enzyme for a variety of reasons. So that stands for ribulose 1,5-bisphosphate, because it acts on, RUBP. So ribulose 1,5-bisphosphate, but then to finish it off, you have carboxylase oxygenase. So that whole thing, that's its big name. So they abbreviate it down to Rubisco. That's just its abbreviation. So the C and the O on the word Rubisco stands for carboxylase oxygenase. So ASE on the end, it's an enzyme, but it's a multifunctional enzyme. It is able to carboxylate. It's a carboxylase. It's able to oxygenate. So that's an interesting enzyme. It can do both. And that has some serious implications for how the plant works with this enzyme. It is the most abundant protein in the world when you look at all the organisms out there, all the way down to microbes that can do photosynthesis, all the way up to the giant redwoods. We have Rubisco acting on the RUBP and the CO2. They estimate about 40% of the protein in leaves of terrestrial plants is Rubisco. Please, right now, as fast as you can, memorize that Rubisco is the enzyme. I've had students mix up and they'll say RUBP is the enzyme. RUBP is the sugar that reacts with CO2. Rubisco, if you can just remember, CO stands for carboxylase oxygenase, ASE, that's the enzyme. Rubisco is the enzyme. Rubisco is the enzyme. Please, 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 please don't mix that up. It's so sad when I read that on that essay. So, okay, so get that going in your head. So this, if you're chemistry minded, this is what it looks like. So you have RUBP here on the left. So bisphosphate, there's 2 phosphates on the 1 and 5 end. Here's your carboxylation process. The CO2 is coming in. They've labeled it so they can track where it actually ends up. And then that's your 6-carbon unstable molecule, quickly breaks down into 2 molecules of PGA. Okay, so now we have, that's it for carboxylation. We're done. All right, we've done our carboxylation step. Do you see anywhere in this reaction RUBP and CO2 are reacting? Rubisco, you have to imagine little Rubisco sitting there where it lets RUBP and CO2 into its active site. Nowhere in this chemical reaction is ATP being shown. Nowhere is NADPH being shown. So that means if everything's going okay and the plant takes CO2 in and RUBP is present and Rubisco is present, this is all just going to happen. All right, it's beautiful. Unfortunately, it doesn't always work so beautifully, but we'll get into that later. And then the next step after that, after you get your PGA, is you have to reduce PGA. So now we get into the reduction phase, but I guess we're out of time. So I'm going to stop there. We're going to get into the reduction phase next week, I guess, and then kind of put it all together on Monday. We'll get into that. All right, so we'll stop there. I tried to tell it to stop sharing.
Yes.